Good morning and hi everybody. Today's lecture is based on heterokaryosis phenomena. So we can saw in the first slide that the heterokaryosis in fungi. So what is the use of heterokaryosis and how it can be present in the fungi? You can saw here in the first, this is the heading of the topic. In the second slide, we can explain about the heterokaryosis. It is actually the coexistence of genetically different nuclei in cytoplasm continuity with one another. Coexistence of genetically different nuclei whose strains are of different. Second, this phenomena was discovered by Hansen and Smith in 1932 in Botrytis cinerea. It plays an important role, that is variability and sexuality in fungi. Actually, the heterokaryosis is the main source of variation in the atmospheric fungi, where sexual reproduction is absent. This term heterokaryosis was proposed by, as I told you previously, Hansen and Smith in 1932, who reported it for the first time in Botrytis cinerea. पहले आपको बताई जा चुका है, the distribution या habit and habitat or the occurrence. It is the most common in fungi but also occurs in slime molds. This happens because the nuclei in the plasmodium form are the product of many pairwise fusions between amoeboid haploid individuals. When genetically divergent nuclei come together in the plasmodium form, characters have been shown to emerge. However, genetic homogeneity among fusion amoeboid serves to maintain the multicellular plasmodium. Here we can see that the genetically different strains of nuclei, as because of the plasmolysis, they come closer to each other. The formation of heterokaryosis, there are so many conditions arises just because that this heterokaryosis is to be takes place. One is the mutation, second is the anastomosis, third one is the inclusion of dissimilar nuclei in spores after meiosis in heterothallic fungi. We can take one by one. That is the mutation. A high frequency of mutation is characteristic of fungi, main source of variability. Anastomosis, that is the fusion of hyphae, yani the two different hyphae that come closer to each other and they fuses with one another. It is mostly intraspecific. Now, when the mycelium or the hyphae they come closer to each other, next step, the nuclear migration from point of fusion to the remainder of the mycelium takes place. Yani that mycelium is to be called as heterokaryotic mycelium. Example, development of heterokaryon in Basidiomycota. As you remember that in case of class Basidiomycetes, either they may be the Puccinia. You can saw here the two different monokaryotic mycelium. They come closer to each other. Ultimately, their fusion, which are of opposite strain, one is of plus strain and one is of minus strain. They come closer to each other and ultimately their nuclei fuses or they makes a pairing with each other to forming a डाइकैरियोटिक माइसीलियम यानी डाइकैरियोटाइजेशन हो जाता है. तो उसमें पर सेल एक प्लस स्ट्रेन का होता है न्यूक्लियर और एक माइनस स्ट्रेन का होता है. In this slide, inclusions of dissimilar nuclei in spores after meiosis in heterothallic fungi. Meiosis results in the production of genetically different nuclei sharing common cytoplasm. Examples are the neurospora, podospora, etc. On germination, they give rise to heterokaryotic thallus. After germination, they develop heterokaryotic thallus. In the asexual phase, occurs frequently in multinucleate spores. You can saw here in this slide, first of all, these are of the two homokaryon haploid mycelium, You can saw here in this, one is of plus strain and one is of minus strain. Ultimately they come closer to each other. इनमें ये फ्यूज़न होता है और जो मेल न्यूक्लियाई हैं दे पासेस टू द फीमेल वन. अल्टीमेटली यहाँ पे न्यूक्लियर पेयरिंग हो जाती है. तो पर फिलामेंट के अंदर आप ये देख रहे हैं कि जो फ्यूज़न न्यूक्लियाई होते हैं ये डिप्लॉयड होते हैं. वन इज़ ऑफ प्लस स्ट्रेन और माइनस ऑफ माइनस स्ट्रेन, और बाकी जो ब्लैक आप देख रहे हैं ये जो है हैप्लॉइड न्यूक्लियाई है. नाउ रिकॉम्बिनेशन जीनोटाइप्स होता है इसके अंदर, और आप ये देख रहे हैं कि प्लस स्ट्रेन और माइनस स्ट्रेन के जो माइसीलियम में मेयोसिस होता है, और ये प्लस और माइनस स्ट्रेन के अलग अलग माइसीलियम बन जाते हैं जो कि ऊपर फिर इसी तरीके से साइकिल में चले जाते हैं. सिग्निफिकेंस ऑफ द हेट्रोकैरियोसिस. द सब्स्टिट्यूट फॉर हेट्रोजाइगोसिटी एंड वेरिएबिलिटी. फर्स्ट, हेट्रोकैरियोसिस एंड पैथोजेनेसिटी. This is very common in case of the rust and smut. Origin of new rays and initial steps in parasexual cycle. These are some important significance of heterokaryosis. Now, establishment of heterokaryosis. The presence of haploid nuclei of dissimilar genotypes in the same cytoplasm. Prerequisite for recombination. This heterokaryosis is brought about by कैसे कैसे कंप्लीट होता है? बाय म्यूटेशन, एनास्टोमोसिस एंड इंक्लूजन ऑफ डिसिमिलर न्यूक्लियाई इन स्पोर्स आफ्टर मेयोसिस इन हेटेरोथैलिक फंजाई. Formation of heterozygous diploid. Nuclear fusion in heterokaryotic somatic cells was first noted by Roper in 1952. It was in Aspergillus nidulans. The nuclear fusion between dissimilar nuclei, the formation of heterozygous diploid nuclei or zygotes, a rare event occurring at the rate of 1 in a million. The heterozygous diploid nuclei fairly stable. The diploid colonies are recognized by either higher DNA content of their nuclei, the bigger size of their conidia, and certain phenotypic characteristics of their colony. The prolonged diploid phase involves repeated nuclear division, enhance the chances of mitotic crossing over. So this is all about the heterokaryosis. This PPT is prepared by Gaurav under my guidance. Thank you.